All right, this is your lecture video for your Chapter 4 Cellular Metabolism lecture. In this one, we're going to be getting into a little bit of DNA synthesis, protein synthesis, as well as reactions within the cell, producing energy within the cell. So if you aren't really comfortable with the Chapter 2 and Chapter 3 material, go back and review that first before you continue on with this video. And even then, maybe go find a crash course or YouTube videos for Crash Course or Amoeba Sisters, something like that, just because they'll give you a broad overview of cellular metabolism. And make sure that you have reviewed your notes a little bit, go through your lecture, your textbook, and just kind of breeze through that chapter, just so you're familiar with a lot of these terms I'm going to throw at you. I'm also going to highlight the APR application that we have on Connect. If you go up to the module and then down to Cells and Chemistry, and then right at the top they have this animation tab. If you click on that animation tab, it's going to give you a lot of videos about these processes we're going through, as well as some from chapters 3. There's some transport processes there, some videos for that. But there's videos on enzymes, there's the translation, transcription, and a whole kind of compilated video on protein synthesis. So after you've watched the lecture video, maybe go through and watch these too, just so that way it can help you visualize these processes a little bit easier. But I will say, they tend to go pretty fast and they are very vocabulary heavy. So you can't just watch these cold and expect this to kind of help you grab it. If you haven't learned or done a little bit of work first, these will probably go over your head. All right, so let's get into this chapter. So first, just remembering that we're kind of talking about energy here right at the beginning of this one, and all of these things are occurring within a cell. So enzyme catalyzed metabolic reactions that maintain the life of the cell and the body. Enzyme catalyzed means that the enzymes, those protein molecules, are what kick off this reactions. The reactions would occur anyway; they would just happen way too slowly to actually maintain life. But enzymes allow these reactions to proceed fast enough to maintain life. And we'll get into what metabolic reactions are, but they are what are essentially supplying our body with energy and making new structures. Our body needs a constant supply of energy for that. We're always growing and repairing structures, and so we have this constant need for energy. Same thing, just transporting things across the cell wall, the cell membrane. Those active transport reactions always need energy. Information from DNA is required to construct proteins, including those enzymes that are going to be catalyzing these reactions. And as I said, metabolism is a group of reactions. It's not just one thing. A lot of times people tend to, kind of colloquially talk about our metabolism slowing down or speeding up, or, oh, they're young, they have a fast metabolism. We'll get into, in these next couple of slides, what actually metabolism is. But energy is derived from the chemical bonds. The energy that's from these metabolic reactions is derived from chemical bonds and nutrient molecules obtained in the diet. So the food that you eat becomes the energy that fuels your body. That's why you can eat things like potato chips and Snickers and Mountain Dew, and your body will be able to break those molecules down and produce energy at a reasonable rate. You can live like that for decades. Some people, live like that for a long time. But really what you're missing out on is some of that optimizing the processes. By not having fresh nutrients, we'll see vitamins and minerals are things called coenzymes that help these reactions to proceed even faster and more optimized. And so you can live on a diet that is very much just kind of heavily processed foods, but by having fresh foods that are varied, you get the variety of nutrients that you need to really make sure these processes are optimized. So looking at the two types of metabolic reactions, the two general categories are catabolism and anabolism. Catabolism is when you break things down. I always think like catabolism, catastrophe, big explosion or whatever, some large, building just breaking down. That's just what goes on how my brain goes. But this is releasing both energy and heat. So 40% of the energy lost, or the 40% of those bonds being broken, are going to turn into energy. We'll look at that. That's ATP molecules. And the rest is heat. And so think about your car. When you're driving in your car, it's it's breaking down the gasoline, and it's turning that into the energy to fuel your car, to fuel the engine to keep things running. But also your car gets hot. You see the oil start to heat up, and you can put your hand on the hood and you can feel the car is hot. And so we also have coolant systems in our car to make sure it doesn't overheat. And so heat is a waste product. In our cars, it really is not used at all, right? We use the energy from combustion, and then the heat is something that we have to deal with, and we have to try to make sure our cars don't overheat. In our body, this is actually what produces our body heat. This heat that's lost in all of these, constant trillions and trillions of catabolic reactions are actually what give our body heat. Anabolism is the other way around. You're making larger molecules from smaller ones. So this is when we're doing things like taking monomers and creating polymers. Right? That's, I think, a chapter 2 topic, looking at maybe amino acid monomers and turning them into polypeptides, your protein polymers. And so if any of those terms were kind of going over your head, go ahead and pause this video, review that, because we're about to jump into it a little bit more. A lot of times these are often reversible, so while you make one molecule, so you're making a larger protein, you can then break that protein down if you need to, and potentially recycle those molecules to be able to then form a new protein. And so this is a cycle of reactions that are occurring. Typically, like I said, they're coupled. The energy that you're forming or releasing rather in catabolism and catabolic reactions is then used in your anabolic reactions. So here's kind of an overview of that. You can see at the top, catabolic large molecules being broken down into smaller ones. It's releasing energy. That energy is in the form of heat that circulates through our bloodstream and kind of warms up our body, and then also towards the actual functional energy that we can use, whether that is active transport or these anabolic reactions of forming larger things for growth and repair. So starting off looking a little bit more detailed into anabolism. So anabolism provides the biochemicals for growth and repair. Like I was saying, things like your triglycerides, your larger proteins, your macromolecules. And anabolism is going to create those. A lot of times that happens through dehydration synthesis. So dehydration synthesis is a type of anabolic reaction. Let's break those two words down. First, dehydration, removing water. So we're removing water, right? And then synthesis is creating something. So we're removing water to join two smaller molecules together, such as two monomers joining together to form that larger polymer. The example they give right here are talking about two monosaccharides, two simple sugars. You remove a water molecule from them, and then they form this disaccharide. Something important to note is that they're not removing a water molecule from each monosaccharide. It's one molecule total between both monosaccharides. So you remove a hydrogen from one and a hydroxyl group from the other. Those two then combine to form a molecule of water, and the two sugars get bound together. Those bonds that they're freeing up will join together with each other, and so that's why they call it a dehydration synthesis. You can that's again a common pathway for making a lot of our other polymers. To make a polysaccharide, like I said, we do two monosaccharides together to form triglycerides, your fats. You're combining glycerol and 3 fatty acid tails, removing water from them, and then with your amino acids, right? You take 2 amino acids, put them together for this dipeptide, and then form dipeptides together to make a polypeptide, and so that's your protein. And lastly, you have your nucleotides joined together to form nucleic acids. These are your DNA and your RNA molecules, and nucleotides are the monomers for those. These next couple of slides are just figure descriptions of that. So you can see dehydration synthesis here going to the right. You're removing this water molecule from these two monosaccharides to create a disaccharide and that free-floating water. So going to the right is your anabolic, going to your left is your catabolic, that hydrolysis reaction. So you can see the double-ended arrow, so it means that it is reversible, but going one way, it's dehydration synthesis, and then to break these two things apart, you add this water, you hydrate it, and it splits it up. It's kind of like dissolving sugar in water, right? You're hydrating those sugar molecules, they'll start to split up and they dissolve in the water. This one is for your triglyceride molecule. You have your glycerol backbone with the 3 fatty acid tails, and you're removing these water molecules here to form this fat molecule. And this one is your protein synthesis. You have this hydroxide on the carboxyl end, this hydrogen molecule on your amine end. You remove both of those and you form this dipeptide bond. So our catabolic reactions, those are then breaking those larger molecules into smaller ones, breaking them back down into their building blocks. So if you're eating a protein source, let's just say chicken. So you're eating a piece of chicken. Your body does not then just take that chicken, that protein molecules from the chicken and absorb that completely. Your body's going to break that protein. It's going to break that substrate down completely into its monomer categories, down completely into its amino acid components, and then absorb the amino acids and then reassemble it into full proteins inside of your cells. You don't just absorb the whole protein, even though the proteins in chicken and our proteins are the same thing. Same thing with like proteins from other sources, vegetarian and vegan sources. It's all still the same kind of protein. But our bodies break it down into monomer components. Same thing with fat, same thing with sugars, and as I said here, and nucleic acids. So catabolic reactions are hydrolysis reactions. If we dehydrate to create new things, we can hydrate those things to break them apart. So you're adding a water molecule to break up chemical bonds. This is responsible for digestion of, as we said, carbohydrates, lipids, and proteins. Sugar, a disaccharide plus water breaks it up into those two monosaccharide components. These tend to be reversible reactions. So even though this one is just kind of showing a one way arrow here, if you were to remove that water, it would then go back into sucrose. These two molecules would join together and form sucrose. So controlling these metabolic reactions, the rate of control is you can have it under enzyme action. So either you are limiting your substrate level, meaning you don't have enough glucose. Let's just kind of stick with the glucose. You're not eating enough sugar. You're not getting enough those that source material, and so your reaction will be slowed because of that, or it can be slowed because of the action of enzymes. So enzymes generally, let's just assume for this first part that you have, or for this these examples, that you are getting enough of that substrate. So that's not the issue here. So enzymes will control the rates of all the metabolic reactions of the cell. That is because our temperature is too low, our body temperature is too low for things to occur optimally. Usually it's about, like, I think 102 to 105 degrees are where these reactions occur at their highest level, which that would put us in the hospital. For most adults, by the time we get to about 104, you can start being at the risk of brain damage, and 105, you can start being at the risk of brain damage. And so for most of us, we would have already been medicated by this point. And so enzymes allow us to have and maintain this base level body temperature, but still these reactions to proceed fast enough. Enzymes, as I said, are proteins that function to lower what's called the activation energy of a reaction so that it can begin and proceed faster. The activation energy is the energy that you have to kind of put in before a reaction actually happens. We'll see that with substrates, that means that when a substrate, let's say it's a sucrose molecule that we were just looking at as examples, the sucrose molecule will bind with your enzyme, and your enzyme will kind of just loosen it up for you. It's like if you're trying to open a jar, and you might kind of run the jar under some hot water and bang the lid on the counter just a little bit, and then that kind of loosens it up, so that way when you actually go to physically twist it, you're not having to put in all this energy. You can just twist it a little and it'll pop right off. You lowered the activation energy to be able to open that jar. And so enzymes do that. They lower the activation energy so that these reactions can proceed really fast, and that's why we have them classified as catalysts. They work in small quantities, and they're not used up in chemical reactions, so they are recycled. So after an enzyme kind of, again, binds with a substrate and it opens a little, or it changes a little bit, so that way the reaction can be catalyzed, the substrate gets unbound, and then the enzyme can bind another substrate, and it just does that over and over and over again, pretty much until it gets a signal to stop. So as I was saying, enzyme bind, and then each enzyme is going to bind a substrate, and it is only acting on that one substrate. It will only act on that one particular molecule. Your enzymes, even though they're recyclable, they are not universal, and so you can't just expect an enzyme from a let's say that's going to be catalyzing carbohydrates to be able to catalyze proteins. They will not happen. They recognize their substrates by their complementary shape. So there's two terms you just need to understand: enzymes and substrates. Enzyme is the protein that's catalyzing the reaction, that's going to be recycled over and over. Substrate is the molecule that you ingested, the protein, the fat, the carbohydrate, and I'm being very basic here because this could also be another hormone. It could be a larger DNA molecule. It could be something different. It does not have to be that. But just since we're talking about chemical react or energy reactions, that's kind of where I was going. So a substrate is what binds to your enzyme. It is what's being changed. And based on the shape of the substrate, that's how your enzyme binds. And that's called your conformation. So the conformation of a substrate of an enzyme is what allows them to latch together. The enzyme has what's called an active site. This is where the substrate binds. So the substrate binds to the enzyme on the active site, and it creates an enzyme-substrate complex. These two things bind very tightly together, and that's why the shape has to be just right. Once they do bind together, though, because we're talking about microscopic molecular structures, it's very, very specific in that shape. Think about how tiny that is. And so when those two form this enzyme substrate complex, because it's these chemical bonds that are coming together to latch them together, that actually changes the whole shape of both enzyme and substrate. So the moment that these two things bind, it's going to shift their shape, and then that causes them to unlatch. The enzyme goes back to its original structure. That's the recycling component of it. But a substrate is going to keep that new shape. That new shape that caused it to dislodge from the enzyme, that's going to maintain its same shape. And so that's lowering that reaction energy. That's how it helps to lower that reaction energy. The speed of enzymatic reactions depends on the number of enzyme and substrate molecules available. So like I was saying, your body can either have a limited number of actual molecules to be able to change, or it could be that your body doesn't have enough enzymes working at that time. At the end of the reaction, the enzyme is released and can be used over by the cell. There's that recycling component. So here's what it looks like in a picture, if you like that. Your enzyme molecule with these two substrate molecules, and then on the enzyme you have the active site. Those substrate molecules bind, and you see these two are now being forced together. Because of the shape here, it's binding so tightly, it's going to cause this binding site between these two substrates to be a little bit different. So this could be our two monosaccharides coming together to form this disaccharide. And you see they form this new molecule. The enzyme remains in its same shape, or returns to its same shape rather. So looking at enzymes and metabolic pathways. A metabolic pathway is characteristic in our body. Typically when it comes to forming energy, it's not just one enzyme, one substrate, boom, there it goes. This right here is a very basic depiction of what happens with enzymes and substrates in terms of its product molecule. Yes, this might be a disaccharide, but this could also be the substrate that then goes into a different enzyme and goes through another reaction, and then that goes through another reaction. And so that's a metabolic pathway. This is a very simple metabolic pathway. We'll look at more complex ones here. So different enzymes control each chemical reaction in that sequence of cellular metabolism, meaning that the product of one enzymatic reaction is going to be the substrate that starts another one. So they're controlling either anabolic or catabolic sequences, and they have to act in a specific order. You have to go 1, 2, 3. You can't go 1, 3, 2. That hopefully just makes sense. That sequence of enzyme control reactions is called a metabolic pathway. And the rate of the pathway is determined by a regulatory enzyme that is just one of the enzymes in the steps. It's going to limit the number of enzyme molecules available. A rate-limiting enzyme, however, is a type of regulatory enzyme that controls the whole metabolic pathway. Typically, it's that first enzyme in a series of reactions. And we'll see how typically the final product of the metabolic pathway is going to inhibit that enzyme, that rate-limiting enzyme. So this figure is showing just kind of the two versions of a metabolic pathway. A is an uninhibited one. So substrate 1 goes through, binds enzyme A, and it comes out as substrate 2. Substrate 2 binds enzyme B, comes out as substrate 3, and so on and so forth. So these aren't brand new substrates that our body is bringing in. These, substrate 2 is the product of this first reaction. And then substrate 3 is the product of substrate 2 enzyme B, and so on and so forth, until you get to your final product. And the second line, we're seeing how the final product here, is the rate limiter for enzyme A. So more product builds up. It's going to start. It could be something like might bind to a portion of enzyme A, not the active site, but a different site on enzyme A, and that causes enzyme A to be inhibited, not be able to bind any more substrate. And then once you don't have this reaction proceeding, where you're forming substrate 2, nothing else will proceed. After a little bit of time, because you're inhibiting this pathway and you're not allowing it to continue, you'll have less product to bind to your enzyme A's. And so then certain enzyme A's, some more and more enzyme A's will be able to proceed this reaction. So it will still proceed somewhat, because unless you have such an overwhelming amount of product that it can inhibit every enzyme A, there's still going to be some of this reaction occurring and proceeding. But like I said, once you kind of get this reaction stopped, your product will also be inhibited. You won't have any more product being produced, and so that will allow the reaction to continue. So it's a self-inhibiting pathway. There's lots of ways pathways can be inhibited, by the way. That's just one of them. So looking at factors that alter enzymes, this kind of goes back to what I was saying right at that title slide, or that first slide about you can survive off of just Snickers and potato chips. I've had friends that do that, and it works. It happens. But you're missing some out on some of these cofactors and coenzymes. And so these alter enzymes to make them function even faster. And some only become active when they have this cofactor bound to them. They can be ions of an element. So remember, an ion is a charged variant of an element. Organic cofactors, things from vitamins or derived from vitamins, are your coenzymes. Something else that you can use to alter enzymes are going to be, because they are proteins, they can be denatured, so they can be unfolded and unwound. And once a protein loses its conformation, it is no longer functional, and that is not reversible. With denaturation, it is an irreversible reaction. So things that can denature proteins, denature enzymes, extreme heat. So even though we said, our bodies do function, or enzymes will function at a higher temperature, once you get to that 104, 105 temperature, it actually might be that that's breaking down those enzymes. And so it inhibits them even more. pH extremes, so either too basic, too acidic, can start to alter their structure as well, because if you remember, pH, acid or base, is just a reflection of the hydrogen concentration, and that's essential for your proteins. Chemicals, electricity, radiation, poisons, as well as other causes can render them non-functional. So there are a range of factors, host factors, that can cause enzymes to not function properly. All right, so looking at energy for metabolic reactions. So energy is the capacity to change something or to do work. Yes, that capacity to change, that ability to change, that having the force, whatever it may be, to shift something or to do something. So types of energy can be heat, light, sound, electrical, mechanical, and chemical energy. So looking at those different ones, kind of think through your brain, maybe some examples of these types of energy, or how you can see these energies in motion or in action. Metabolic reactions use chemical energy. That's typically the common one that we're going to see, where they actually break bonds between molecules and use that energy source. Chemical energy is held in those chemical bonds of molecules and released when the bonds break. So when you break open a bond, that releases a little bit of heat, a little bit of energy, and that can be used. Release of chemical energy often occurs through the oxidation of glucose. So when we're looking at our bodies and how our bodies produce energy, it's typically through this breaking down of glucose in a process called cellular respiration. Oxidation, we'll see oxidation and reduction. You might have heard, or you might remember those reactions are the redox reactions, the same kind of thing. And so oxidation, I want you to think about, and when you learn it in chemistry, you typically learn it about in terms of the transfer of electrons and hydrogens. Since this is a biology class, I want you to think about when you see oxidation, this is referring to breaking something down. So oxidation is a breaking down of glucose. You're breaking it apart. When you see copper out, and my aunt has these lovely birdhouses, and they have these copper roofs, and they start to break down. They patina over time, and they're supposed to, right? That's she thinks it looks nice and pretty, and it does. But that's the oxidation of copper. It's being broken down and causing it to do that. When iron starts to rust, if your bike starts to rust or something because it's being left out in the rain and outside, that's an oxidation of those iron molecules. And so I guess your bike's not iron, but you know what I mean. The when you see those kind of things, that's a breaking down of those molecules. And so our body goes through oxidation of glucose through the cellular respiration process. Cellular respiration releases chemical bond energy when those nutrient molecules, your glucose molecules, are broken down. It can't be used directly. We'll see that there are some cases where a body might, just kind of break down a glucose molecule and use that energy real quick. But generally speaking, to use energy in our body for any type of effective way, it has to be transferred into ATP, adenosine triphosphate. So once you break down these molecules of glucose, and we'll see how that goes step by step, each step along the pathway releases a little bit of energy, and that energy is then stored to create ATP, this adenosine triphosphate, 3 phosphates. About 40% is captured, transferred to those electrons that help make ATP, because when you're forming bonds, we go back to that chemistry real quick. When you're forming bonds, you're talking about transferring electrons. And so electrons are what are holding your energy in your bonds. When we talk about the energy of a chemical bond, it's really the electrons storing that energy. And so by synthesizing ATP, we are binding those electrons together and we're storing it, kind of like you would put energy into a battery before it's released. The other 60% that was not harnessed, as we said, is released as heat. That travels into your bloodstream and then is circulated all throughout your body. So technically, your blood is about a couple, 2 to 3 degrees warmer than your body temperature when you were just to take it, through your mouth or something. Okay, so let's look a little bit more at ATP, our adenosine triphosphate. So we'll see this is an adenine molecule, so it's an adenine nucleotide that has these 3 phosphates. It's been modified to have these attached to it. There are 3 phosphates in a chain. It's the main energy carrying molecule in your cell. It's used for a lot of different functions, active transport, synthesizing different structures. So ATP is crucial, essential for function. It is, however, not the only energy molecule in our body. There are other types of energy molecules that we use depending on the organ that we're talking about or the availability of resources. But just generally, almost 99.88 or 0.9% of the time, it's ATP is your energy molecule. Both the second and the very end terminal, so the very last phosphate bond and the second to last phosphate bonds in the chain of ATP, those are the really 2 high energy ones. So what will happen is that those bonds between those phosphate molecules, that's where the energy is being stored. And so they'll pluck off a phosphate, and when they do that, it releases this big burst of energy, and that can then be harnessed. You're left with then what's called ADP, adenosine diphosphate. What this doesn't show is that you can then also lose that second second phosphate bond, and create AMP, adenosine monophosphate. If your body, it's not as high energy as that terminal one, but it does still have a pretty decent amount of energy stored in there. ADP or AMP can both be converted back into ATP, and they're interconverted through that cellular respiration process. Cellular respiration releasing energy, and reactions in the cell which use the energy from these splitting reactions. So here's a molecule of ATP. So this is your adenosine molecule, and then you have these 3 phosphates, and these wavy lines I think are supposed to represent our nice high energy bonds. So you can lose this one, you can lose this one, but this phosphate will stay bound. Here's another depiction of that, if you like to see it in a kind of graph organization. You can start with your ATP molecule here with these high energy phosphate bonds. But let's say, we need to do something. We need to transport this molecule of glucose into the cell. Oh, okay, well here, I'm going to give you this phosphate. That'll split off. You're left with ADP, but it's no big deal because cellular respiration happens constantly, and there's all these free floating phosphates around. So we'll just add this phosphate back to you, and boop, right there, you're back at ATP, and it can continue on and on. This is showing that cellular respiration process, just very generally, starting with glucose and oxygen, C6H12O6, right? That's glucose plus an oxygen molecule. You go through your catabolic reaction. I'll let you tell me what kind of reaction this is. Is this a hydrolysis or a dehydration synthesis reaction occurring along this arrow? Correct. This is a catabolic reaction, so this is a hydrolysis reaction. And then you have this anabolic reaction where you're taking the energy released from this one, from splitting up all these glucose molecules into ATP by synthesizing it. All right, so now let's look at cellular respiration and kind of these processes a little bit more in detail. We'll go ahead and say that your notes, this textbook refers to 3 steps of cellular respiration, and some you will see 4 steps. It's just an intermediate phase, and I'll point that out to you in our notes where that will happen if you look at other resources. But for this one, we talk about cellular respiration having 3 reactions in which glucose is catabolized, so broken down to carbon dioxide, water, and a little bit of energy. The 3 reactions are glycolysis, citric acid cycle, and the electron transport chain. So glycolysis, let this lysis inform you that it means splitting up. So glycolysis is the splitting up of glucose. Citric acid cycle, we'll see this is a cycle, so it's a constantly recurring recycling process. And the electron transport chain. So this is where we're actually going to have this chain, this little metabolic pathway, this connection of enzymes that are moving electrons from one to another, and in doing so, harnessing that energy that's being released through the rest of the pathways. These are stepwise pathways, meaning that one has to occur before the other can. The product of one is the substrate of the other. And again, with the citric acid, it's a cycle, so the product eventually reforms into the original substrate. Yeah. And then this is just kind of summarizing that reaction again. You have your products, your substrates on this side, glucose and oxygen, and you're forming carbon dioxide, water, and energy. Here it is, just kind of seeing a metabolic cycle. So you have this enzymatic reaction, this first reaction that starts with restores your substrate 1, whatever your base product is, your end product from the pathway up here continues down into here. It goes through a reaction to restore substrate 1, and then you have that same kind of stepwise path where the product becomes the product of one reaction is then the substrate for the other, until you end with this final product that gets probably released, and then you have again some new substrate coming down the line. Glycolysis is the first reaction in cellular respiration. It's the first thing that happens. We're literally splitting the glucose. So we're taking a 6-carbon sugar and turning it into a 3-carbon molecule of pyruvic acid. And so you do not have to learn them, and be grateful if you ever get into biochemistry. You will. Any higher level chemistries, you'll have to probably learn that. But for us, just understanding that you're taking glucose, which is 6 carbons, and splitting it into 2 3-carbon pyruvic acids. This phase does not require oxygen. It's an anaerobic function, so anaerobic without oxygen. So it will occur whether you are just sitting at your desk, breathing just fine, or you're at the end of a marathon and you're about to pass out and you can barely breathe. Glycolysis still occurs. It occurs in the cytosol. This does not happen in the mitochondria. The other 2 steps will happen in the mitochondria. It does take some energy. It takes some ATP to split apart this glucose. But ultimately, even from this one step of glycolysis, there's a net gain of ATP. You do actually get 2 ATP out of this. So even though you're starting with glucose, the products are going to be 2 pyruvic acid molecules, 2 ATP, and some high energy electrons. If you've studied cellular respiration before, this would be, usually we would talk about NADH and FADH here. Those are just electron transporters. For this though, we're just talking about high energy electrons. But again, they're bound to something, right? They're usually bound to a hydrogen that's kind of carrying those electrons along. After you've made pyruvic acid, 2 pathways are available. If there is no oxygen present, so again, you're kind of, pushed yourself, you aren't getting enough oxygen in to be able to funnel these aerobic reactions, it's going to go into lactic acid. So your anaerobic pathway for pyruvic acid is to convert into lactic acid. This gives you very, very short periods of energy. It is not very effective as an energy molecule, and it will not be very sustainable. If you've ever run or pushed yourself hard enough, worked out hard enough that you start to get muscle cramps and you have to stop and you just truly cannot function anymore, it's because you're in this lactic acid pathway. I will go ahead and say you're not sore the next day after a workout because of a buildup of lactic acid. As soon as lactic acid is produced, within seconds to minutes, your body is breaking it down. So soreness comes from actually damaging your muscle fibers, your muscle cells, not because you have a buildup of lactic acid. So just saying that. If you do have oxygen present, which is going to be most of the time throughout your life, throughout your existence, then pyruvic acid is going to enter the aerobic respiration pathway and continue on with those citric acid cycle and electron transport chain. So aerobic with oxygen, this is where it's going to be your highest energy reaction, metabolic pathway. So again, these are pretty dense slides. We'll get some pictures here in a couple of slides, but if this stuff starts to get overwhelming, go and look back at your textbook if you need to, review some notes, watch some videos, or do that after this. But if these phrases are overwhelming, if these terms are overwhelming, I understand. You might have to look at this a couple of times before it starts to click. So when you do have, in the presence of oxygen for aerobic respiration, that pyruvic acid, those molecules, those 3 carbon molecules that were formed from splitting glucose, are going to enter into the mitochondria and start the aerobic respiration pathways. These won't continue without oxygen. It first starts with the citric acid cycle and then goes to the ETC. So it's in between. It's right as pyruvic acid. It says it will lose a carbon atom. This is that intermediate phase that I was talking about, because pyruvic acid has to be converted a little bit into what's called acetyl-CoA, acetyl coenzyme A, before it can really enter the citric acid cycle. And so some text will highlight that as its own 4th phase in this metabolic pathway. We're not looking at it though. We're just kind of saying, oh yeah, it just gets converted really quick, and then acetyl-CoA from the pyruvic acid, that's what enters your citric acid cycle. The citric acid cycle goes through. It takes this. It's still a 3 carbon molecule. It's just got some other stuff attached to it. So it's taking that. It's going to be splitting it more and more, getting rid of oxygen and carbon. You can see here. And it's going to release energy as it does that, but really it's these high energy electrons. You're getting, again, 2 ATP from here, you're going to get 2 ATP from glycolysis. That's just a little byproduct. Really what we're forming are these high energy electron bonds. Because those are what then go into the electron transport chain. It uses those high energy bonds and essentially passes them from one to the next, and through what we call oxidative, until we go through oxidative phosphorylation. And that's a big word, that's a big phrase, so I understand if it's like, wait, what? It feels like we just skipped a whole bunch of stuff. And we did, thankfully, because there are multiple steps. I think there's 5 or 6 steps along the electron transport chain with enzymes at each one, different substrates at each one, and so you don't have to learn all this. So if it feels like this is a very kind of like, well, just kind of starting and stopping, yes. Sometimes it does help to hear those little intermediate places, again, to kind of highlight those individual enzymes and substrates, but it's even more overwhelming to learn. So again, we're starting kind of up here with glycolysis. That gives us pyruvic acid. Pyruvic acid is converted into acetyl-CoA. Acetyl-CoA goes into the citric acid cycle, and it's just churning out, some carbon dioxide, but really this ATP and these high energy bonds. Those high energy bonds are gone, then get put through this electron transport chain where they're just passed from one to the next, one to the next, one to the next. But each time they do that, they release a little bit of energy that can then be harnessed. And that energy is then used to phosphorylate ADP to ATP. So let's go back to this oxidative phosphorylation. We said oxidation, oxidative, this is breaking things down. Phosphorylation is adding a phosphate to something. So if something is phosphorylated, phosphate, phosphorylation is adding a phosphate to something. And so what this phrase here, oxidative phosphorylation, means is that you are using the energy let's start with this one. You are adding a phosphate to something by breaking something else down. And in this case, we're breaking down all of those high energy bonds that we've been forming along this pathway and using that energy to then add a phosphate to ADP to make ATP. The last oxygen acceptor, because there's just always those extra little oxygen electrons that are kind of floating around. The last one is oxygen, and that forms water. So that's why water is there. This little phrase, oxygen is the last electron acceptor, it sounds so important. It sounds so kind of intense. I don't know why. It will stick out if you, as long as you're studying biology and you have to study cellular respiration. Oxygen is the last electron acceptor. It's just one of those phrases they love to say and love to add in. Um, if it comes up on a quiz, if it comes up on a worksheet, test, or whatever, there's that phrase. Is that maybe it'll stick out to you now that I've kind of highlighted it. But really, just remembering that you form water as a byproduct in cellular respiration. Water, carbon dioxide, energy. And that energy is in the form of these 32 ATP. This is at optimal level. Our bodies aren't always working at optimal level. Sometimes we don't have, again, those proper coenzymes, those proper nutrient vitamin factors to be able to reach this optimal level of ATP. Sometimes the structures that we're using, the enzymes that we're using are older and they don't work quite as well. And so, 28 to 32 ATP, 26 to 32 ATP, that's usually about normal. So yeah, just that's the number that we're introducing here. 32 is the number that you should remember. But when it comes to actually thinking about how the bodies work in practice, they are not always going to be doing 32. That's why it says up to 32. So this was the metabolism of glucose. This was the oxidation of glucose. This all three steps. Started with glucose, that was our beginning product, and we're ending with these ATP. But you can also oxidize proteins and fats through aerobic respiration. There are certain diets where people severely limit their carbohydrate intake, which can be dangerous because your brain does not use ATP. Your brain does use the breakdown of glucose. And so when you don't have enough glucose, then your brain does not function properly because it doesn't have the energy it needs. But proteins and fats can also be used in situations as needed. We'll look at that in a minute. Here is a graphic depiction of the respiration of glucose, the cellular respiration of glucose. You can see up here you're starting with this 6-carbon, those are little carbon molecules, the 6-carbon glucose. As it goes through glycolysis, breaking that glucose up, you get 2 pyruvic acids. These each enter 2 different cycles of the citric acid cycle. You have 2 different citric acid cycles happening at the same time to process both of these pyruvic acids. Remember, though, it gets converted. Pyruvic acid gets converted into acetyl-CoA. And then as it goes through this citric acid cycle, you can see you're losing some carbon dioxide, you're losing some ATP here, and then there's some more of those high-energy electrons. I didn't highlight them up there either. Those high-energy electrons at each of these little pathways, those get funneled into the electron transport chain. So electron transport chain, this is what's going to produce your ATP. This says 28, but if you add, I know we said 22, 32, 28 plus 2 is 30, plus 2 is 32. The other products for your electron transport chain, of course, are that oxygen. Those leftover hydrogens, electrons, oxygen molecules create a water molecule. And so that's just kind of that pathway drawn out for you. So here is showing how we can actually also use other molecules to be able to funnel through this metabolism pathway. So this is just food. This is just normal, this is a breakfast that is being represented. I don't even say breakfast. This is some food that is being represented. You have proteins, carbohydrates, and fats. Proteins from those egg whites, carbohydrates from the toast and the bread, and then butter is their fats. So these can all be broken down into their monomer components. Proteins into amino acids, carbs into simple sugars, your glucose, and then fats into glycerol and fatty acids. And then each of these can enter the metabolic pathways at different points. Glucose, your carbohydrates, will obviously go right through glycolysis, but your glycerol molecules can be converted into pyruvic acid. And these fatty acids and the amino acids can be converted into acetyl coenzymes. And so that allows them to enter at different points. It's not as effective. It's not as energy rich as glucose is going to be. Glucose is going to be our highest energy kind of molecule for this pathway. But things like your fats and your proteins can also provide energy for you. Okay, so now let's get into a little bit more of some other synthesis reactions. So now that we know how we create energy, let's look at uh, DNA a little bit, because that is then going to dictate how we use that energy. Once we have the energy available, what are we going to do with it? So DNA is your deoxyribonucleic acid. Notice that prefix of deoxy, it means that it's lacking in oxygen. So we'll look at RNA, your ribonucleic acid, DNA, deoxyribonucleic. DNA has your genetic code, the instructions needed for synthesis of proteins and processes required by the cell. So talking about that a little bit more, your DNA is this strand. It has 3 billion base pairs long, I think. So 3 billion little code, 3 billion codes, lines along in this entire DNA chain. Each section that actually has the information for making a protein is called a gene. So you might have heard of genes on your cells, or genes on your chromosomes, genes within your DNA. That's what this is. A gene is just a section of your entire DNA molecule that's going to code for a single protein. And you have genes all along your body. That's what scientists kind of work to try and find is, well, where's the gene? Where's the section of DNA that regulates for making this protein, that regulates for this allergic reaction, that regulates for this disease development, this disease marker development? How can we start to maybe monitor, or in some cases start to alter and splice out those genes, and that maybe would then reduce the chances for, let's say it was like the BRCA2, the breast cancer gene. They could be able to remove that, or cause that to be inoperable if they can find that gene and then isolate it and then remove it, or like I said, somehow cause it to terminate, then we could reduce a lot of the cases of breast cancer. The complete set is called your genome, right? So they sequenced the genome, the entire human genome, back in like the, I think, early 2000s. And it was a very big deal because now we knew kind of the whole sequence. But just because we know the sequence, just because we know what all the letters are, doesn't mean that we can read it. Reading it that's how we isolate the different genes. So genes are telling us specifically, oh, this codes for that. Genome just gives us the whole text. The portion of the genome that encodes proteins is the exome. Everything else is just for regulatory processes, or not used at all. It's just kind of space markers to make sure that other genes can be coded properly. So DNA consists of building blocks called nucleotides. So the monomer of a DNA molecule, of a DNA polymer, is your nucleotides. They form two sugar phosphate backbones with the bases pointing to the inside. And then it twists. It has that helix shape to it. I'm going to jump ahead just one slide so you can see that. So this kind of bluish purple, I'm going to say blue, lilac maybe, periwinkle, this, it doesn't matter. This kind of ribbon on the outside, that is your sugar backbone. That's your sugar phosphate backbone. And you can see there's two of them. They're intertwined. This twisting shape is called the helix. And then on the inside of the helix, this is where you have these base pairs that are being paired together. A and T, C and G. Okay, with the bases, there we go. So right here, the bases found in DNA nucleotides are adenine, guanine, cytosine, and thymine. A, G, C, and T. We'll see that in RNA, it's slightly different. But they're joined together by these weak hydrogen bonds. Weak because that allows them to be split open for replication and protein synthesis, and then form back together without it being a big deal. The nucleotides of one DNA strand are complementary base paired to the other. So A only binds with thymine, adenine only with thymine, and cytosine only with guanine. That makes sure that they're lined up together properly. And then, as we said, it twists to form that double helix. And here you can see it. So during cell division, each new cell must be provided with an exact replica of the parent cell's DNA. Replication occurs during interphase, so hopefully you remember that from chapter 3, that cell cycle phase where during interphase you have the genetic material replicating, duplicating to prepare for the cell to be split. The DNA helix separates by breaking those hydrogen bonds between the base pairs, between the A and T and C and Gs. The twisting unwinds, and one strand will come apart. One strand kind of breaks off, and that is your coding strand. DNA polymerase is an enzyme. So anytime you see ASE, that refers to an enzyme. So DNA polymerase, polymer. So this is an enzyme that is going to join a bunch of things together, specifically DNA molecules. So DNA polymerase joins new nucleotides to each strand, forming complementary pairs to the original strands. So as those strands are broken open, this kind of unwinding of the helix, it allows DNA polymerase to go in there and it will pull base pairs kind of from the cytoplasm, and it will just boop, 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 boop. It will add them to this complementary, to each of these complementary original strands to form two new strands of DNA that can then be split. So each daughter cell in a cell division has a copy of the parent cell DNA. So essentially you end up with one parental strand and one newly synthesized strand. This is one of those places where, or a big place where mutations can start to occur and issues can start to develop. The more often that your cells have to divide, so the more often that your cells are exposed to damage, radiation, pressure, getting, even something just simple as getting cut and scraped a lot. All of those things, exposure to different chemicals, can cause your cells to constantly have to go under cell division at a more rapid rate. The more rapid that has to happen, the more chances there are for issues in this replication process, and then the more chances for things like cancerous cells to develop. So here is seeing that process. You have this original DNA molecule that gets split open, and then DNA polymerase is going to come in here and it's going to find different base pairs to be added on, and they form these new double helix strands. Okay, so then protein synthesis. Like we said, DNA is going to be coding for our protein synthesis. It supplies the genetic instructions for that. The genetic code is a sequence of base pairs in a gene that specifies the order of amino acids to make a particular protein. Okay, so there's a lot there. Genetic code is the sequence of base pairs, so your A, T, C, and G, in a gene that specifies the order of amino acids to make a particular protein. So our body is going to read these base pairs. It's going to figure out which base pair is being coded for, which amino acid is being coded for based off of the base pairs, and those amino acids get joined together and eventually make a protein, dipeptides, polypeptide proteins. An amino acid is represented by a specific sequence of 3 nucleotides. We call it a codon on your DNA. So a specific sequence of 3 codons, 3 nucleotides. 3 nucleotides is a codon. Is going to code for one amino acid. Many amino acids come together to form a protein. There are also start and stop messages. The genetic code also has nucleotide sequences for start. That's always AUG. I always think school starts in August, AUG, start codon, as well as stop messages for the genes. The process of protein synthesis is both transcription and translation. Transcription is creating a readable source, a readable template that we'll see is called mRNA, and then translation is turning that template into protein. It's translating it into a protein. So for transcription, DNA molecules can't leave the nucleus. They are bound within that nuclear envelope, but protein synthesis happens in the cytoplasm with the ribosomes. And so there has to be a way to get this template strand out into the cytoplasm. And it does that by using RNA. RNA is that ribonucleic acid, so it has an oxygen present where deoxy would not. It's a little bit shorter, and it can leave shorter than DNA, and it can leave the nucleus. It has a couple of different modifications. It is single-stranded, so it doesn't have that helix shape. It has ribose other than deoxyribose. And one of the base pairs is different. Instead of thymine that we have in DNA, it uses uracil. So our T switches to U. This means that adenine will bind to uracil when we're talking about RNA molecules versus thymine. Still the same kind of order, but just something that you have to think about is that, oh yeah, it's going to be AU for RNA instead of AT. And that's also how one of those ways you can quickly identify if you forget about something like the single-stranded, or you don't see the ribose sugar. If you see a uracil instead of a thymine, you know you're in RNA. And that can happen. You're like, well, when would that happen? If it was just written out, if someone would just get to give you a written codons or written genetic strand, and it had uracil instead of thymine, that's how you would know. This creating a transcription, this creating this RNA strand is called messenger RNA, mRNA. And we'll see that there are multiple types of RNA. There's also transfer RNA, ribosomal RNA. But this particular one, this transcription strand, is your mRNA. And it's a messenger, so it's bringing this message of the genetic strand out into the cytoplasm for the body. That whole process is called transcription. RNA polymerase, then, so looking at how this is made a little bit more, thank goodness. So the enzyme RNA polymerase is directing the synthesis of mRNA. We said DNA polymerase helps with DNA replication. RNA polymerase, so there's that same polymerase making an enzyme that synthesizes, putting a lot of things together. Making this RNA polymerase is still using the rules of complementary base pairing, still that A and U this time, and C and G. It recognizes the beginning and end of a particular gene in which DNA strand is the proper one to copy, because if it were to copy the non-coding strand, then you would get basically the inverse of the protein that you need. So the RNA polymerase is able to figure out which one do I need to bind with based on the way the chemical bonds are lined up, and that's the way that it reads the strand to be transcribed. When it reaches the end of the gene, it releases the new mRNA strand, and that completes the transcription process. So RNA polymerase is this enzyme. It travels down your DNA strand, and as it travels down the DNA strand, it's polymerizing, so it's adding all of these RNA nucleotides together to transcribe this mRNA strand. Here's a comparison of RNA and DNA molecules. You should definitely look through that and know this information. Same thing with the complementary base pairs. Make sure you understand how they pair, how they bond to each other, and what the difference between DNA and RNA is. So now translation. Now that we've gotten our messenger RNA, we've got this strand that we can actually turn into proteins. How does that happen? And this happens through the process of translation, translating mRNA. So as I said, each amino acid, which is your protein monomer, is going to correspond to a 3-base sequence of DNA called a codon. A codon. So that codon is going to be what is going to be the template. So we have one codon that represents the start message at the beginning of the gene, and 3 different stop messages. So a codon will only have one of these 3, but we'll see that there are 3 different kind of combinations of base sequences, base nucleotides that means stop. Whereas there's only the one start, AUG. And hopefully when I say AUG, you are understanding, oh yeah, adenine, uracil, guanine. mRNA comes out of the nucleus and finds a ribosome in the cytoplasm. That's where the protein will be constructed using that nucleotide sequence. In the cytoplasm, another type of RNA called transfer RNA, your tRNA, has a 3-base sequence of nucleotides called the anticodon, which is complementary to the messenger RNA codon. So your messenger RNA has the translated strand. Your tRNA, each tRNA molecule, and I think I'm going to just jump ahead a little bit here. I know this looks really confusing, but let's just talk about it. So this is the genetic code. These are your codon charts. So these are all of the different amino acids that our body has, and then this is how they are sequenced. So let's start with that AUG. So that's the start codon. So it's right there. You can go ahead and see it. We can go ahead and jump right to that. It's methionine. That's the amino acid. So every amino acid chain is going to start with methionine. But to get to the amino acid, we first have to start by reading the RNA. So the first letter in our RNA chain is A. The second letter is U. So there's U. So we're in this box. And then the third letter is G. So it's AUG. Okay. So that is our mRNA strand. That's going to be our mRNA strand. The tRNA molecule will have the opposite of that. So it would have UAC on the tRNA. But attached to that tRNA molecule, we have to come back here. Attached to this tRNA molecule, on the other end of it, is that methionine amino acid. So every tRNA molecule on one end, it has this anticodon, this 3-base pairing that we just looked at. And then on the other end of it, it has the amino acid that's being coded for. That tRNA molecule will go down the mRNA and it will boom, boom, boom. As that mRNA slides through the ribosome, it will add the proper amino acid chains together. So the ribosome holds the mRNA in position while the tRNA carries in the correct amino acid in sequence, anticodons matching up to codons. There's 20 different types of amino acids, different types of tRNA for each amino acid. Some amino acids are coded by multiple anticodons. That's just normal. Ribosome consists of rRNA and protein. So you have 3 different types of RNA that we've been introduced to now. mRNA is your messenger RNA that comes that actually has a sequence for the transcribed sequence. tRNA transfers amino acids that are appropriately bound to your mRNA or appropriate reflecting your mRNA. And then rRNA is your ribosomal RNA, and that actually creates the structure for processing translation. Between the rRNA and other protein structures, your mRNA and tRNA are funneled through that, and then that's what allows amino acid molecules to be attached to each other. Again, this can be a little tricky if you're not used to visualizing it, so that's why I very much encourage you to go and watch some videos either on Connect or on your own, find some others for you to watch to be able to envision this process. And then maybe come back and watch this again and see if, oh yeah, okay, now it makes more sense. So the ribosomes contain enzymes that help join amino acids together by, hey, there's our friend dehydration synthesis. So as this rRNA, this ribosome is being sliding down your mRNA strand, it's allowing tRNA to bring in amino acids, and then the ribosome is joining those amino acids together. It has those enzymes in there that allow the amino acids to get joined together, and then your tRNA gets kind of shunted off to go and find another amino acid for it to bind to and bring in. As the amino acids get joined together, the new protein molecule folds into that particular conformation. So that helps it to actually be completed through that translation process. Once it's complete, mRNA, tRNA, and ribosomes can all be used over again. So these can all be, an mRNA strand can be translated many, 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 many times, and it's typically being translated by multiple ribosomes simultaneously. Typically you have multiple ribosomes running down your mRNA strand because you never just make one protein. Your body's going to make a whole bunch of it. And so this can just be done over and over and over again. Again, there's that chart. So if you're ever trying to figure out what amino acid you're being coded for, you look at the first, then the second, and then the third letter, and it will tell you, is it lysine, is it proline, serine, tyrosine, whatever it might be. Those are your 3, by the way, stop codons, UAA, UAG, and UGA, just in case you want to keep those in your brain. Here's what that process looks like from your DNA turning into messenger RNA, that leaving the nucleus, being translated by finding the amino acids attached to your T. See, it looks like almost like a T, like a cross. Here's your anticodon, there's your amino acid, and you see the anticodon matches up with the mRNA strand. When the mRNA strand matches up, it brings this tRNA and this amino acid together. And then as the ribosome moves down your mRNA strand, it's just going to link. It's going to bring all these tRNA molecules together. They deposit their amino acid, and then once that's been deposited, the tRNA can jump off, and then you form this polypeptide chain. Here is just more protein. It's essentially the same thing. So it's just giving you, truly it is the same thing as this. So it's just kind of giving you a different image of that. So read through both of these, see one might have, say it slightly differently than the other image, and it might help you understand a little better. So translation is finished when the protein has finished folding. So correct folding is required for the functioning of the protein and the maintenance of good health. If our proteins can't fold properly, it could be one of those issues of temperature is too high, pH is being thrown off, chemical factors, radiation factors, that kind of thing. So a protein being able to fold is a protein that can function. If a protein isn't able to fold correctly, so maybe there was an issue or an error in that mRNA or tRNA sequence, because as you see, there's a lot of different spots that can happen. Maybe it was a DNA replication error. But if one of those errors kind of occurs and is not caught and is not corrected, the protein won't fold correctly. So they can either be refolded, maybe there's some shifting that can happen, or completely destroyed by proteasomes. A gene that's been transcribed, translated, and is a functional protein is said to be expressed. Gene expression is the basis for cell differentiation. So the fact that your liver and your kidney cells and your toe cells all act differently is because they're expressing different genes. And those genes are particular to those cells' function. All right, that's your whole lecture. Reach out if you have any questions. Thank you so much.